Hey, Riffee, again, coming at you live from my kitchen again. But I'm going to talk about Unit 2, cells and tissue, particularly just cells. Here we go. So, there's a particular branch of biology, and as you know, biology is the study of life, but there's all these very specific branches within biology or very specific segments of biology that study very specific things. The one that studies cells is called cytology. Cytology simply studies cells. We'll get into what the study of tissue is later, but cells are based on this one solid theory that is called cell theory, and that comes from cells are the building blocks of all plants or animals, or basically cells are the building blocks of life. So by building blocks, meaning you start with one, you add another, you add more, you add more, boom, you get a plant or an animal. It's unbelievable. So if you check it out, you take cells to make tissue, to make an organ system. You have several organ systems, boom, you get an entire organism. Building blocks. You build upon the individual cell, you get more and more, boom, you get an organism. Second thing, they're the smallest functioning units of life. You can either exist as a multicellular organism or a unicellular organism, but regardless of the fact whether you're made from one cell or millions upon billions of cells, you are considered alive, and so therefore we consider a cell to be the smallest functioning unit of life. You can think about viruses. Viruses are not composed of cells. Therefore, we do not consider viruses to be alive, even though they are the most obnoxious thing on the face of the planet. They give you the cold, they give you the flu. I hate them, don't want them. I'm glad they're not considered alive. Okay? Ooh, a unicellular organism. I believe that's an amoeba. One cell, it's still alive. Amoebas wreak havoc. I believe there was an article just recently about amoebas and the fact that a guy wore his contacts or slept in them for like months upon months, and he actually had an amoeba eating away at his eyeball. It's intense. Okay. The third part of the cell theory is that cells are produced through the division of pre-existing cells. That's that whole chicken before the egg concept, meaning which came first. So think about it this. Human beings came from pre-existing cells. Mom, dad, come together, sperm meets egg, fertilized, boom. Sperm is a cell, egg is a cell, pre-existing cells. They come together, and through the division of those pre-existing cells, you get a new human being. So life occurs from pre-existing cells. So where did the cells start? Check it out. Fertilization, and you get division upon division upon division upon division, boom, until you get a human being. But starts with two cells, half the genetic information combining to form one person. It's amazing. Miracle of life. Okay? Each cell maintains homeostasis. So the cell itself maintains a functioning, working internal environment, which then has this cascading effect called interdependency upon every biological level above it. So as long as a cell maintains homeostasis within itself, or all cells, then the tissue those cells are functioning with should be fine, which causes the organ to be fine, which causes the organ systems to be fine, which then you get a fully functioning organism. Okay. This then goes into that whole interdependency concept, and if you, from my videos, have not figured out that I've been drowning interdependency into you forever, since we've started this class, it's a big concept. But we're going to now dive into more the organelle level. Inside cells, there are organelles, which are small, independent, functioning, specialized structures within a cell that each do their own job. And as long as they do their own job, the homeostasis within the cell is maintained, which then the cell maintains homeostasis, then the tissue, and so on and so on and so on and so on. But what we end up with also is there's this level of interdependency, and in order to cause a human being to, or allow a human being to function properly and more efficiently, we have specialization of cells. That means that certain segments of the DNA have been turned on or off and actually cause the cells to have a very specific job. Think of it like an assembly line. You have one person put tires on, you have one person that put the steering wheel on, you have all these different people, and it's faster to be good at one job and more efficient to be good at one job than it is to try to be good at building an entire car. That's how your body works. You have very specific things that are in charge of very specific jobs, and your body works more efficiently and faster if those tasks are divided up evenly. It's not necessarily evenly, but divided up to where it works faster and more efficiently. Examples of the diversity of human body cells based on the fact that their function affects their form. Remember that whole anatomy physiology relationship. So here's some examples. So what we have here, red blood cells. Completely different shape, completely different structure. You also have some white blood cells in here. Those things are shape and function are related to each other. They serve a very specific job. Then we have muscle cells. These are smooth muscle cells. If you look at these things, they have a completely different shape than red blood cells. But the reason they have that shape is because they serve a specific function and they have one function or job. Then you have, this is bone tissue or bone cells. Each one of these individual small little black dots has bone cells within them. They form what are called osteons. This looks completely different than the previous two examples because it serves a specific function. Then you have epithelial cells. You probably aren't going to like epithelial cells in tissue, but we're going to get into that. These things protect or are exposed to an external environment. If you notice here, there's nothing here, but there's a whole bunch here. These cells serve very specific functions in hollow tubes within your body. Your skin is epithelial cells. We'll get into those really hardcore here shortly. These are adipose. These are adipose cells or fat cells. Again, look completely different. Here's all the fat reservoir, and here's all the cellular functions. So their job is to store and process and use fat for fuel. You have a sperm and you have an egg. Even the two within themselves, completely different. Look at how much bigger an ovum is than a sperm cell. But on top of that, they serve two separate functions, and their function is related to their shape. Tail, for I guess you could say locomotion. Large egg containing all nutrients and things necessary to cause rapid division and fertilization and the incorporation of foreign DNA into the blah, 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 blah. I think you get the cell diversity thing. Cell membrane. I'm going to nail cell membrane later, so I don't really want to get too much into it, but it's the boundary of the cell. It creates physical isolation. It separates what's inside the cell from what's outside the cell because you want the inside to function well, homeostasis, and you want it to be separated from the outside because sometimes the outside area outside of that cell is a toxic environment that can affect the functioning of the inside. So you want to create physical isolation. The other thing you want to do is the cell membrane regulates what goes in and out. These are ions, nutrients, waste, secretions, excretions, all of these things controlled by cells at the cellular level. You also have sensitivity. These things, cells can maintain the ability to detect environmental stimuli or environmental changes. This is where we get into that whole receptor and things like that, but receptors are made from cells. You also have structural support. So the way a cell can be arranged or in its function can actually provide support. In particular, the cell membrane, it provides a support for whatever's inside the cell and maintains the environment from outside the cell. You also have those membrane proteins that are embedded in there. There's several different kinds. You have receptors, channels, carrier proteins, enzymes, anchors, identifiers. There's tons of them in your book labeled 3-2. You don't have to know specific ones, but later on in part 4 of our notes, we're really going to get into receptors, channel proteins, and carrier proteins when we talk about things moving inside and out of a cell. We'll get into that later. Okay. In order to transport materials across the cell membrane, there's a whole bunch of different various methods that are used. Again, there's a table in your book, table 3.3, that outlines some of these various methods to get things in and out, but I cover them more in depth in part 4, so we're going to tackle that topic there. Another structure of a cell that is really important is actually the cytoskeleton. This is what provides a protein internal framework that actually gives the internal structure to a cell. Think of it like the frame of a house. You look at a house, you see the outside, you go in the inside, you see the inside of the house, but within those walls is the framework that actually provides strength and flexibility for that house. Cells have a similar thing, it's called the cytoskeleton. Microvilli, cilia, and flagella. So we're kind of going through all these kind of different cellular structures that probably haven't been dove into as much as in your other biology classes, but In human anatomy, microvilli, cilia, and flagella, more microvilli and cilia, play a huge role in maintaining homeostasis within the body. Microvilli are these small, very small, finger-like shaped projections that extend out of the cell membrane and are actually exposed to the outside surface of the cell. Those extensions are supported or connected to the cytoskeleton. So the cytoskeleton provides the framework for actually microvilli also. If you look at this picture, these are epithelial cells, and I believe these are possibly cells of your trachea. Yeah, of your trachea. What these are the cells in this hair-looking, this is kind of not a very good picture, but it's the best one I could find on short notice. These are all of the cilia that project outside of the cell, and they serve a function, which we're going to get into here in just a second. Cilia are longer, slender extensions of the cell membrane that are also supported by the cytoskeleton. So microvilli are small, cilia are longer. Okay, cilia are the hair-like projections that line your bronchial tubes. Their biggest job is they're extensions of the cell membrane, but they try to trap microbes, debris, pathogens, things that don't need to be entering your lungs. They try to trap those also within the mucous membrane, and then they actually create a flow that moves up from the lungs, up your trachea, into your mouth, and then you swallow mucus, saliva, or in extreme cases when you have like bronchitis and you have that hardcore cough and you're hacking up phlegm, that's because there's crud in there that you don't need, and your body's trying to get it out. So the cilia trap it in the mucus, and this cough happens, which its purpose is actually a positive purpose. Even though it hurts, you're trying to hack up stuff that's not supposed to be in there, that's trapped by the cilia in the mucus. Okay, so here's things both microvilli and cilia do. I would pay attention to this part. You might be tested over it. Here we go. First thing to do is they increase the surface area for absorption. The more finger-like projections you have, the more surface area there is for things to attach to them. So that allows for more attachment points. The other thing they do is it allows for more absorption of materials and for the secretion of stuff. So the more area you have to put stuff out or to absorb stuff, the better. It actually creates a current or movement. So if you think of them like this, if my hands, this portion of my hand is the cell, and my fingers are the cilia or microvilli, when they move, they can create a flow in one direction, which can cause things to travel along that flow or current or movement. That's how stuff gets out of your lungs, how you hack stuff up, how constantly you have this swelling of mucus throughout the day. They also provide structures to trap foreign bodies. We'll get into later. Flagella. Really don't see them anywhere. They resemble cilia, but they're much longer, usually existing as a single structure. You will see this with sperm cells. You will see this in single-cell organisms that will have flagella in order to project themselves or create locomotion through whatever environment they're living in. So, for example, here's a single-celled organism which has a flagella. Here's a sperm cell that has one. Here's the ovum. That is the actual act of fertilization. So, let's get into the nucleus. The nucleus, I hate the analogy of the brain of the cell because it doesn't do any thinking. What it does, however, is it has the instructions for how the rest of the cell is supposed to be perform, or how all the other stuff is performed. It is the largest organelle. When you stain any kind of cell and look at it under a microscope, immediately one of the most notable structures is in fact the nucleus. So it is the control center, and by control center I simply mean that it controls or obtains or holds, or however you want to do it, the genetic code which is used for the synthesis of proteins. I know in biology we went over transcription, translation, and all that jazz, so I'm not going to dive super deep into it like we did before. But you still need to know how we go from nucleus within the DNA to the way you look, the way you act, the ability to process lactose. All of these things comes down to the genetic code within one single cell or within all your cells. So let's talk about that real quick. The DNA or genetic code inside of each of those individual cells, and through cell differentiation and specialization, actually some portions of the DNA get turned on and some get turned off. And what actually happens is you get a very fine-tuned function. Those specific things, sections of DNA that are turned on, are used to create proteins. That is called protein synthesis. So let's talk about protein synthesis. You know it has two steps. It has transcription, where you take DNA, you split it in half. You use one half of that DNA and you make an M messenger RNA copy of DNA. That mRNA is then used as the instructions for assembling a protein. In order to assemble a protein, mRNA is used as the blueprint for the sequence of amino acids that are used. When several amino acids get put together in a row, you get a protein. But before you can assemble those, transcription is the making of an mRNA copy of DNA. After transcription occurs, you get translation. This is where the mRNA leaves the nucleus, goes out into the cytoplasm, attaches to a ribosome. That ribosome then uses transfer RNA or tRNA to take amino acids, bring them in, assemble them into a chain, a long chain. Once that chain becomes long enough and you reach the stop codon, that chain breaks off and you have a long sequence of amino acids, which is called a protein. That protein then folds to a very specific shape based on the sequence of amino acids. That shape of the protein and the sequence of amino acids then determines how the protein will be used or what it can do. Those proteins then are used within the cell. Keep in mind, the sequence of amino acids and the shape of that protein determine the function. There's like something like 40,000 proteins within the human body that can be created. So, when those proteins are used within the cell, their specific organization or line of amino acids and the shape are used in a chemical reaction, and that different shape and order of amino acids determines what happens at the end of the chemical reaction. So what you end up with is protein function is determined by the sequence of amino acids and the shape of the protein. Let's say DNA or RNA for that matter, whether it's mRNA, has something go wrong where maybe a single letter is changed or anything, those things are altered. What that then does, by changing one single amino acid, you have the probability of changing the entire, if you change one specific nucleotide of DNA, you have the probability of then changing the instructions for how to make a protein. So changing one nucleotide or one sequence or one letter of DNA can cause the entire sequence to be altered in terms of amino acids. So if you change DNA, you then change RNA. If you change RNA, you change the sequence of amino acids. If the amino acid sequence is changed, the protein is changed, whether it be in terms of sequence of amino acids or structure or folding shape, which then if that protein is changed, you alter how it is used in a chemical reaction, which then destroys homeostasis within the cell, and all hell breaks loose. Okay? I told you that was a fast transcription translation. You might want to review that and go back through it, but there's also other organelles. See this list over here to the right-hand side? Those are a list of organelles that you need to know their function. I'm not going to go super deep into those, largely because it has been pounded into your brain in the last 3 biology courses you've ever had in your entire academic career, and it's a really simple 2-page thing in your book where you just kind of cytoplasm function, centrioles function. So make sure you understand those things pretty well. Cell diversity and differentiation. So let's get into the cell diversity and differentiation thing. We talked at the beginning of this about cell diversity, meaning there's this wide range of diverse amount or types or look or cells within the body that their form is related to function. All the somatic cells, keep in mind that somatic cells means body cells, not sex cells, that make up an individual have the exact same chromosomes, exact same genes, exact same sequences of DNA within them. However, you end up with these things that are liver cells, fat cells, nerve cells, muscle cells, red blood cells that look completely different, even though inside of them they have the exact same DNA and the exact same chromosomes. So how does that happen? You have the same DNA and the same instructions. How do they end up with different jobs? That's when we get into diversity and differentiation. How cells become different in terms of their roles is basically comes down to specific genes in the DNA sequence are turned off. That then deactivates that cell's ability to make certain proteins. So by turning off all these different sections, you're left with only one section that is on. Therefore, that particular cell can only perform one task, which makes it very specialized and different from all the rest. As more and more genes throughout the process after fertilization occurs and as those cell divisions are going over and over and over again in the womb of mom, as more and more genes are turned off, the cell becomes more restricted on its abilities and thus specialized on what it can do. That is called differentiation. After fertilization and several cell divisions, differentiation begins and produces cells with very limited capabilities, but don't take limited capabilities as a negative thing, take it as a positive thing. They can do very, very, very specific tasks. These cells that form a very specific task and are grouped together to form a very specific function form tissue. Can I get some oohs and ahs? You're probably saying ooh and ah at home. Okay. Now let's talk about how people grow. You can grow in a couple different ways. Here's one. You can either change the size of the cell, which basically means a cell can get bigger or it can get smaller. When a cell increases in size, not number of cells, increases in size of cells, that is called hypertrophy. So, if you increase the cell size, you increase tissue size. Hypertrophy is what occurs when you increase the size of your muscles. You don't get more muscle, you have the same amount of muscle, just those cells get bigger. So, you could look at it this way. When you're born, you actually have a set amount of muscle cells, and as you grow, you actually are more so increasing the size of those muscle cells. Comprende? Atrophy is the opposite of hypertrophy. This is the decrease in individual cells, thus decreasing the size of the tissue. You ever heard the whole "you don't use it, you lose it" phrase? Well, this is where atrophy comes into play. A lot of people that have had casts on their arms, they don't use those muscles in their hands or wrists or things like that, and what that results in, a decreased use in the muscle, decreases the size of the muscle. When that cast is removed, you actually end up with smaller muscle mass. Not less muscle, not less cells, just smaller muscle cells. Here's an example. Normal bicep, atrophy. This guy could have been put in a cast, not using his bicep, boom, shrinks in size. Atrophy. Okay. You can also grow in terms of changing the cell number. This is really simple. You make more cells, they take up more space, you get bigger. Hyperplasia is the increase in cell division, which results in an increase in cell number, which makes the tissue bigger. It's a response to changes in your body's internal environment, homeostasis, which can be normal or abnormal, actually. Normal, example would be increase the number of milk-producing cells during pregnancy. Abnormal would be increasing the number of cells due to uncontrolled cell division. This is cancer, which results in a tumor. So if you increase the number of cells, because they're dividing so rapidly, you get a tumor. That is the abnormal growth. Here's examples. Normal cells, hypertrophy, hyperplasia. So if you notice, there's only 4 cells here that are much larger. Each individual cell is grown, but there's more cells here, but they all take up the same amount of space. So you can grow by hyperplasia, which is increase in cell number, or by hypertrophy, which is increase in cell size. Here is abnormal cell growth. Unfortunately, this would be a brain tumor. So let's get into tumors. Tumors are also called neoplasms. They are called by abnormal hyperplasia or new growth of cells, and they may or may not be harmful, believe it or not. Benign tumors are the harmless ones. They actually can be removed and they don't alter the function of other body parts or neighboring cells. This is a benign tumor. As gross as it may be, it's not altering the function of neighboring cells, but however, it is altering the homeostasis of her entire body, because when you have a tumor on your lip like this, it becomes very hard to eat. Once it's removed, boom, good to go. Then you have malignant tumors. Malignant tumors are the not good ones. These are harmful and usually can result in a fatal form of hyperplasia. The tumor or the cells develop a abnormal function, which then causes them to divide rapidly, and that rapid dividing causes an influx or an increase in the amount of cells that are in that area. Well, what they actually start doing is they start robbing neighboring cells of things they need to survive. So the cancer cells start taking over in like stealing and taking all the nutrients. And that's what results in a decreased homeostasis. But another thing that can happen is also called metastasis. Metastasis is when a portion of a tumor breaks off and travels throughout the body, and it can actually result in cancer in another portion of the body. Brains, tumors. Okay. Unfortunately, I put all your entrance card questions at the end of this one. So I'm going to click through these slides real quick. I'm going to let you read them, let you pause, let you rewind the video to go back to different things, let you process what's happening. But you have 5 entrance card questions that are due to me. They will be for a grade. I'm going to click through those slides real quick so you can do that. Pause at each one if you want. Other than that, I love you. Thanks for watching the video. I hope it helped. Let me know if you have any questions. You know how to get a hold of me.